So with that, we've already corresponded over email, but I'll introduce myself. I'm Ben. I'm one of the application scientists at DeNovix. I've been here for almost 3 years now, and I got my master's at Villanova in immunology, so I do have a lot of background in cell counting. So I'm going to start with the CellDrop and just kind of focus on that first. So anyway, just to give you a quick background on DeNovix, we have been around for just over 10 years now. I believe our 10-year anniversary was in January. So a lot of the original company started out at NanoDrop before it was purchased. So we have a lot of experience making instruments, making devices that are reliable and trustworthy. Our first product was the DS11, which I'll get into next, but the CellDrop has been a huge product for us, even though it's young. So with that, I'll just hop into that and point out some things and show you how the instrument works. So I don't know how much experience you have with other cell counters, but most of them require plastic slides or some kind of plastic that's disposable that you load the sample into for measurement, and then you either throw it out afterwards, or it can sometimes be glass that you wipe clean, but those can also crack and have to be purchased fresh. The really nice thing about our device is we have an arm. So we'll show you, I'll point that out real quick. This arm is has a piece of sapphire on it here, and there's another piece of sapphire here. So what we can do is, instead of needing disposable plastics, we can create a more precise chamber with pieces of glass. And then there's no disposables. There's nothing that's really at risk of breaking. So it's a really low cost of operation instrument once you purchase it. Just to point out all the different features we have on the device. We have, your regular bright field counting for just, if you want just cell counts, no viability. We offer Trypan blue counting, which is a very popular bright field viability counting program. We also have fluorescent viability counting, like AOPI. So I'll get into that a little more later, but that's a combination of 2 fluorescent dyes, acridine orange and rubidium iodide. And that's a really excellent improvement to the data you get from Trypan blue. AO and PI alone are a little less commonly used, but they are used, so we include separate applications for those. And we also have a GFP application to assess transfection efficiency. I can cover that as well. I have a GFP sample here to show you. We have a yeast app. That app will not count in bright field. It's to use our FDA and PI yeast viability stain. And then we have primary cell versions of the Trypan Blue app and the AOPI app. And those are just really better default settings to get you started when you're using a primary sample like PBMCs or whole blood. If this didn't address everything you would like, we offer custom methods so you can kind of create your own, choose your own adventure, if you will, on the CellDrop. You can make your own assessment if none of these are satisfactory. And then we have your regular, housekeeping apps. We have a user guide for the device just installed right here. That can also be found on our website. We have a calculator. It does regular calculations, but also volumetric dilutions as well as cell dilution. So it can be actually a bit handy. The settings app covers all the Wi-Fi and all that kind of stuff. We have 21 CFR capability. We have user accounts, so different users. I know this is particularly useful at your group, is different labs can create pass protected user accounts in different apps, so they can keep all of their data and protocols segmented off from each other and don't have to worry about another group editing them. We store all the data on the device in this data app here, and that it's on a 120 gigabyte SSD inside the device, but it's accessed through this data app. And last but not least, we have the updater app. So over time, as we add features and improve the performance of the CellDrop, we make those updates freely available to all our customers, so there's never a charge to update the software. With that, I think I'll plan to just hop into Trypan Blue and cover some of the major features of the CellDrop as we go. So just like any touchscreen, you just click on things to open them. So while I'm here, I'm going to grab my sample, but I will just mention that the ability to load our cell samples without a slide is a patented technology through DeNovix called direct pipette. And it allows for more speed and accuracy on the cell count because plastics tend to bend and warp, and every manufacturing procedure has tolerances. So when you're using a really precise motor and optical grade sapphire that tends to be very consistent, you don't have to worry about some of the additional features that have to be built in to accommodate for or overcome, imperfections in a plastic slide. So the direct pipette technology is really critical to why we feel we're a much better cell counter. So the way you load a sample is what I've been doing off screen. I don't think you can really see me, but I have a tube of cells mixed 1-to-1 with Trypan blue. I pipetted several times to mix because it's a very small volume. And what I'm going to do to load is I'm going to put the pipette tip. I'm going to try to keep my glove out of the shot. Pipette tip in the groove here. There's a groove cut into the sapphire. I'm going to slide the tip all the way up the groove so that it's actually touching the sapphire surface on the arm, the upper surface. Once it's there, I know the tip's all the way into the groove, and then that's when I want to load my sample. So I just simply pipette, and then on the next screen, you can see a live preview. So we have a little junk in this sample, but that's okay. Otherwise, it's relatively clean. For anyone who's curious, these are HEK 293T cells, so a very commonly used cell line here. So what we have here is 10 microliters, and we tell you on the count button what you need to load. So the standard for most of the plastic slide competitors, as well as the very traditional glass hemocytometer, you're going to load 10 microliters, and it's a 100 micron chamber. So we're simulating that here with the arm. We've made a 100 micron chamber, and we're loading 10 microliters in. And that's good for most concentrations. But because we have this arm and we're not fixed to any manufactured product, we can actually, using the software and the motor, we can raise or lower the arm to accommodate a wider range of concentrations than our competitors are capable of at a 100 micron height. So you can go to a 400 micron height. You have to load 40 microliters, but that's great to get our range all the way down to 700 cells per mil, which is really, really tiny. And then we can also decrease the chamber height down to 50 microns. You load 5 microliters, and that's great when you have something that's over 10 million cells per mil, and maybe you don't want to spare a lot of volume. So they're both, and the reason we chose all those heights and concentrations has to do with statistically significant samples, but I can cover a little more detail on that in a minute. This one's already in focus because it's our demo unit, so I will point out that once you've kind of focused the device, it's so consistent, even though it's been off and cleaned and doing all this stuff, it returned to the last used focal position, and it already looks really good. So the way I can tell that is I can zoom in. So just like any phone or iPad or anything like that, you can pinch to zoom. So I like to do that on an area where there's a good amount of cells like this. And then we want to change the focus. And we do that by clicking on that button. And then to check it, we have a guide. So anytime you see, I'm going to point without clicking. So I'm going to point with my pipette. Anytime you see this lowercase I in a circle, it is a info button, and those are clickable throughout the software on the device, and they give you little hints about what you're doing. So in this case, we're showing you 3 examples of focus, 2 that are improper, and 1 that is correct. And what I would say correct focus is actually the bright white center and the sharp dark border on the cells. I don't expect there to be a lot of dead cells. I think that's probably a dead cell, and so is that. But this is a really fresh, healthy sample, so I don't expect a ton of cell death in here. And the live cells, because they're excluding the Trypan blue, should have sharp, dark borders and bright white centers. I will point out now that we do have autofocus. So if you don't want to go through up and down, coarse and fine adjustments, it's not too difficult. We're already quite close. So I think a couple fine steps like that, I think that's probably fine. But we can also run the autofocus just to show you how simple that is. And this is, all the current cell drops that are leaving the factory are coming with this. This was highly requested, so we were able to go through and add it to the software. You can see it doesn't take all that long to run. And once it's done, I don't think it adjusted my focal position all that much, maybe a few steps. But I'm going to trust what the computer found with autofocus. And that's really it. The other thing to check with Trypan Blue is the exposure. This exposure looks good. And the way I can tell is by looking at again, my guide. Overexposed washes out the borders. Underexposed kind of conceals the borders because everything's too dark. And the correctly exposed, the middle ground is you can see all those cell structure and all the borders, but you don't wash anything out or lose it in the darkness. We have two exposures. We have a normal and a low. Normal is typically good for 1:1 in 0.4% Trypan, and low is good for 1:4 in 0.4% Trypan. And if you ever want to make custom contrast-based dye exposures, we have a custom exposure wizard and guide on here. I won't walk through it now, but it's possible to make any other exposure repertoire that you could need. Once you've done that, I think it's good practice to name your sample so you can keep track of it. I'm just going to call it 293T. You could put the description, maybe if there was a drug treatment or you want to keep track of what you did. Then a passage number. I think this was like passage 30, so we can put that on there too. And that'll all be tied to each sample. And I'm just going to count with default right now, just so you can see how it'll work. And we'll press count. You'll see that the count actually proceeds quite quickly, only a couple seconds. And we're done. And now we're on this screen going to indicate live cells with green circles and red cells with dead cells with red circles. If you zoom in, just like on the last image, you can see exactly what the CellDrop called. And as you're getting used to it and trying to learn how the CellDrop works, it's really important to go through what I'm doing right now and actually go through and see if I was hand counting this sample, do I agree with how this is deciding to call each object? So is everything that's being circled red, dead, and is everything that's being circled green alive? Do I agree with that based on my sample? There's a lot of neat tricks in here once you're on this screen, so I can zoom in on a little area. And let's say I don't want to count real big things like that. So you can gate in real time. If you grab down here and drag it, you'll see the circles disappearing as I go past. So I'm setting an upper limit. I've dropped it down from 30 to 19, and you saw that circle disappear in real time. And you're also seeing the math over here changing in real time as I do that. So I'll slide it back up so you can see, but the numbers change as I make adjustments. You can do it the same way with bringing the minimum up. If you don't want to do it looking at the picture, you can pull up a histogram and do it this way. So if you say, based on a technique I'm doing, I can't have anything larger than 25, you could set a gate that way. I'm not going to, but that's something you could do. There's a few things in here that I might disagree with. So maybe that guy and that little circle here. So there's ways to optimize those out. So what I did, just to emphasize that, is there's an optimize settings button. So I just use the default protocol for this, and not that the default protocol won't be sufficient for everyone's cell type. So it's important to go through, like I said, when you're first using it and confirm the count. So what I might do here is increase my dead roundness to 30 just to get rid of that lighter cell, and maybe I'll even increase this to 40. So I want to get rid of some of those lighter objects. I might increase this to 60 just to get rid of some of the light objects as well. And then I'll hit count with current settings. And the cool thing is this doesn't require it's not going to take a new image. It's just going to reanalyze the old image that was already there. And you can actually see I was able to get rid of that circle and that circle. So I was able to successfully optimize my settings that I'm actually happier with. So that looks a lot better to me. And now I don't have to write this down in a lab notebook and bring a Post-it note with me every time I use the CellDrop. I can save these settings to the protocol. I can make a new protocol with these settings. And I'm going to call it HEK293T. So I recommend using the name of the cell line to keep track of your protocol so that you know what it is for next time you want to use it. So now that I'm done, I'll press next count. You see that already it's going to use those settings that I just created for the next count because it's assuming you might want to do more than one with that sample. And this can store a ton of different protocols. So you see this is our demo unit. We have a lot of protocols that have built up over the course of running some demos. And we have a whole list of unique protocols saved on this device to be, loaded for different samples. If I click up where it says general, we have all of these different, again, test accounts that have been made because this is our demo, but you can have all these accounts be pass protected. So if I wanted to log into Dave's account, which I'm not going to do because I don't know his password, my protocol that I just created won't show up in there, which is really nice. It's a nice way to keep everything separate. So if you create the protocol in your account, it'll only be present in your account in that application. Yeah, so now I'm going to point out how to do a cleaning. So you can see the sample's still here. This is our live preview. So I hit next count, our sample's still sitting on the glass. So it's time to wipe that away to move on with another sample. So the way you do that is actually quite simple. You just lift the arm. And then from there, you take a Kim wipe. And I like to fold it up in a nice, neat square like this. I'm trying to position it in front of the camera because it prevents, things tearing off or whatever. And we also don't, there's also a technique for cleaning. So this allows me to present it best. So what we want to do is wipe unidirectionally. So notice how I'm not scrubbing back and forth. We found, especially in the drier winter months, that'll build up a static charge between the glass and interfere with sample loading. So what we recommend is one direction, a few wipes, and very firm pressure is preferred. And they're sapphire glass, so you're not going to scratch it. I've always kind of make a little joke that unless you have diamond infused Kim wipes, you really won't damage the glass. So apply plenty of pressure and clean both the lower surface and the upper surface on the arm. And I recommend bracing the hinges on the arm so that if you're pressing, you don't crack anything while you're doing that. When you set the arm back down, we'll flip back over to the live preview, and you can see that there's really nothing left on the screen, which is excellent. The other good news is the device is very robust. So if there are a few little particles on there, we have found through plenty of research that those particles are flushed out by the next cell loading. But if you're worried about it, if it's been maybe 5 or 10 samples, we generally recommend 70% ethanol cleaning with that. So just loading it the same way you load a sample, letting it sit for a few seconds, and then wiping clean as I just showed you. And that will actually be sufficient to fully clean everything in between, in the midst of a large run. So if you're worried about reproducibility, that's very fine. We also allow 10% bleach, pretty much any chemical solution that won't react poorly with sapphire. So hydrofluoric acid is out, but 70% ethanol, 10% bleach are typically the preferred method. Okay, so if there's no more questions about Trypan Blue, I will move over to our fluorescent counting in AOPI. So you just press the home button in any app to return to the home screen here. And then you just click on the next app when you're ready to use it. So you can see there is a little bit of stuff on there from the last cleaning. I can wipe that off real quick. Sometimes it just requires a little bit of an extra wipe if you missed a spot. But it should be fine. We'll see if it goes away when I load my AOPI. Typically that does go away. And this is a great time for me to point out that the way AOPI works is fluorescent counting. So the way the dye interacts with cells is it requires them to have a nucleus to be stained either green or red. So I highly doubt any of that schmutz has a nucleus, so it'll be ignored regardless. So again, I'm just pipetting up and down to resuspend. 10 microliters again here. And I'm going to load. You can see the sample flow across the screen, and you see that that little patch was flushed away when I loaded a new sample. So that's good news. Okay, so on this screen, we still see our cell sample. We see some different buttons. We see this white button here. So that's actually our channel selector. The white indicates that we're on bright field. The bright field isn't used for counting in this app. So I really just use it as a reference frame to make sure nothing's moving. So what I'll do is go to the green, and you see some nice, pretty signal on this screen. Looks like everything's pretty well exposed here. And then I'll just check our red, and I'll go through exposure in a minute, but I just wanted to point out our two filters real quick. So I'm going to go back to green because these look a little bit off. So I'm going to go down. Yeah, they're definitely a little too bright. 16's high. So what we're doing here, again, like everything else, we show correct, underexposed, overexposed. Essentially, if you've ever done any fluorescent microscopy work, you know that the rule of thumb is minimize the background noise and maximize the real sample signal. So if it looks a little underexposed, but you don't have a lot of background, that's more ideal than having it be nice and bright and having too high of a background. I think this is probably good. The detector is very sensitive, so it'll see just about everything. I'll go to the red and just check on that exposure. Take that down, that's way too high. I generally find that my red and green exposures are within one of each other. So they're either exactly the same or they're within one of each other. I don't find that I have to have, 15 in green and 8 in red or something like that. So just to keep that in mind for your cell sample. And then the last thing to point out is, again, focus is really important. So we have focus in this application, and we recommend you focus in green because we're using green to count. So you always want to focus in the channel you're using to count. So we're counting in green and red, but they're pretty similar. The bright field tends to have a slight offset from them. So what we want to do is use green to focus. Again, I think autofocus is the best call here. Just to highlight that we can do it in all the different applications. It'll pick a really good focal plane. And what it's trying to do is just make sure that there's no noise around the cell border. So we want them to be as round as possible without any bleed over past the real cell border. And that's the goal in both exposure and in focus. So you don't want to overexpose so much that it's bleeding beyond the edges of the cell, and you don't want to focus in a way that blurs the edges of the cell because we want to have an accurate cell count. So I was not in a great focus, and that's why the autofocus failed, but I can just adjust by hand so we don't have to keep sitting here staring at it. It takes a little longer because it has to wait for the exposure on this. So the lower the exposure, the quicker it'll adjust frames. So that looks pretty sharp to me. Fluorescence is very forgiving. So as long as you're pretty close, it's usually sufficient. So again, we're going to name our sample. I'm just going to name it and not worry about the other info. And this one, when you press count, it takes a little bit longer because we're actually taking 3 images. We're going to take a bright field, a green, and a red image. And then we're going to overlay them and count. So we already have our result. And what you see is the overlay. And it's drawing, it's hard to see. So if I zoom in all the way, you can actually see it's drawing a green circle around the green cells, but it does a very good job of finding the border. So it's very tight along there, and it's sometimes hard to see that it actually drew a circle. But it's doing a very good job of seeing the green signal and circling it green, and likewise for the red, circling the red signal and calling it, dead. I'll try to find a good example. So here's an example of maybe wanting to reprocess. So if there's a little additional exposure in the background, sometimes that can cause a problem where it draws these big circles, where the way to get around that is you just reduce the maximum diameter and you don't allow it to draw a really big circle, and then it'll find all the cells like that. So it's quite a simple procedure to do that. And it looks pretty good to me. If you ever see a cell where there's a little bit of red in the background, it probably means the red exposure is just a little too high and maybe it needs to come back down, but you see it's not really impacting the count. If the red exposure is too high, it can tend to bleed through the green filter. So it's not actually that the cell is green and red. It's just sometimes it overwhelms our filter set. Again, we'll create our protocol. We'll save it. Oh, there's already a 293. I'm not going to bother with that. There's already a 293 protocol, so it won't let me overwrite it. But you guys get the gist of how to create a protocol and do all of that. Another cool feature you can do on the CellDrop is we have a dilution calculator. So what it does is it pulls from the live cell count here, and it pops it into the current cell concentration, because we're assuming you want to use live cells for your data analysis. Say you're ready to do flow cytometry, you're aiming for 1 million cells per mil, you want 2 mils in your tube, and say you have 5. You put all that in, and now down here, you actually get a dilution guidance. So it tells you how many mils of your cells to dilute in mils of buffer, and it gives you a really accurate dilution guidance based upon your live cell count. So that can be really helpful. You can also put that in if I hit next count, there are calculators here. So you can put it in before the count is processed. So if you know no matter what the cell count is here, I need it to be this concentration when I'm done, you can just put it in so it saves you some work at the end. Another way to navigate, so right now you can see them on the count tab, you can click on the results tab and go back and forth, but we'll stay here for now. You can look at, if you wanted to look at the data, it's just like green or red, or just green, like you can change all the channels up there. And then the last button I haven't covered, it's in both apps, is the export button. So if you just wanted to export this sample, you can do a CSV, which just be open in any Excel or a program like that. You can get a nice PDF report, which is laid out in a very organized fashion that shows all the parameters. You can have it include images or histograms, which I'll show in a minute. And then you can get all the raw images off the device too as .png files. So if you ever need assistance from DeNovix for a protocol, or you want help, or you want to show us a sample that doesn't look right to you, the best way to do that is to export those images onto a flash drive, or here, I'll show you the options. But the best way to do that is to export the images and send them to us, because it's the most helpful way for us to give you feedback, and we can actually reprocess here and give you better help. There's a USB port. You see that the first option selected is USB. I don't have one with me, but if I had one, I could plug it in the front left. There's also 2 ports in the back, so there's 3 USB ports total on the device. They can just be plugged in and exported simply. You can set up a network printer. So if you wanted to print over the network, as long as the CellDrop is on the same network as the printer, you can do that. You can set up network folders. There's a million ways to export your results. You can also email. So if you had an email account hooked up to the device, you can then email yourself the results if you forgot your thumb drive like I did. So there's a ton of ways to do this. They're all really simple. We try to make it easy, but that's the best way to get your data. Just to show you the graphs that I mentioned. So right now we're in the single sample view. Anytime you see an arrow, a line with an arrow, that would indicate it's a drop-down menu. So if you click it, something pops down. We have a results list. So if I had multiple samples, they'd all show up here. We also have a graphs tab. That's kind of what I was talking about. So here for this sample, we're graphing based on % viability, but there's a bunch of different graphs we can show. We can do a histogram of live dead by size. So we do green and red, and then the number of cells, the cell diameter, excuse me, and then the number of cells with that diameter that are either alive or dead. So we break everything down. There's a lot of ways to visualize the data. If you had multiple samples, you can actually select 5 and have them on the same graph and compare them. So if you wanted to compare your % viability to, on 5 samples, you can do that right here. And those can be exported later. You also can generate these on your own using the CSV. So all this data is pretty much pulled right off the CSV. If you have the CSV with all the samples on it, you can make these graphs yourself as well. But yeah, this is now we're back on the main screen, and we're just going to wipe clean when we're done. Once you've closed an app out, all the results that were in there are moved to this data app. So if you go back to that app, they won't show up in the results screen anymore, but they're safely stored here. When the arm is lifted to clean, the sample's automatically saved to the internal SSD. So you never have to worry about, you notice I've never pressed save or done anything. It's all been saved for me. We make that very easy. We don't want anyone to lose their data. So if I wanted to find, let's say I wanted to look at all the AOPI results on the device, the only thing I have to put in to search for data is I have to put the app in. After that, everything else is optional. So if I wanted to pick a protocol, I could filter by date, all that kind of stuff. I'll hit search, and then you just see all the samples on the device show up. So I'm just going to do select all, which is that button. I'll do add to report. And then you see, again, there's a report tab up in the top. So I'll click that. And we have the list. So I can make this look like the window we were just in by hitting single sample. If I go to list, we see every sample on here. So you can actually click, like that. You can select that way. And then you can export just the ones you've highlighted. So if you hit CSV here, it'll grab 1, 2, 3, 4, 5 samples that I clicked and put them in one CSV document. If you wanted all 78 on one CSV document, you hit select all and then hit CSV, and they'll all be there. So you can do as much manipulation and processing with all the numbers in one file later. The last thing I'll point out briefly, because as you've noticed, the apps all kind of work the same, just with the different features that make them unique apps. I'll just quickly show GFP. So I have a GFP sample here. I'll pull up that app. You notice it looks, again, just like every other app. It's in a bigger tube, so I just vortex it. So I'll just load them on here. We have plenty of cells. And this is a great one for assessing transfection efficiency more than viability. It actually won't give viability data because it's just going to count in bright field and in green. So the focus already looks pretty good here. Just going to check our exposure. It's a little too bright. So I'm going to come down just a bit. And then I'll go back here, I'll count. And I'll just show you the couple differences in this app versus the others. But GFP is a common question. And if you're curious, we have a technical note on our website about how to best set up GFP experiments on the CellDrop and how to blank and do all that, how to set up a negative control. We have a whole stepwise procedure typed up. So if you want to do GFP assessments, I definitely recommend going and looking at that protocol because it's a little more complex than the other stuff. So what you can do is zoom in and you can see that it's identifying bright field stuff in blue and GFP positive stuff with green circles. And you notice we have an additional slider here, which is our intensity gate. So what you can do is determine how much GFP signal is, your floor. So what's your minimum amount of GFP that you want to include? We go into more about how to do this in that note. We set the default to 20 here, which is fine. But you can gate looking at this. So normally you would have one big peak and then pretty flat. This is a cell line that's all expressing GFP. So there's varying levels of GFP. You can see that some of these are just not meeting the threshold and they're therefore excluded, even though they're green. But if I drag this down, you'll see them start to come into, so we're counting them as green now because we're lowering the minimum intensity that needs to be there. And if I slide it up, you see that number, the circles are changing in real time. You also see that we're giving you a % efficiency based on the total cells and the GFP positive cells. But that's really the only major difference for this application versus the others. And again, definitely look at the tech note before you proceed with an experiment here. Based on all that, I think that pretty much sums up what we need to cover for the CellDrop. We talked about data applications. I'll show you real quick just some of the other features we have on here. I don't think most of this is going to be that important for you. If you want to check your storage, which might be helpful since you're going to have a lot of labs using it, you can go to the storage tab. You see that total available is 115 gigabytes, and it actually tells you how much percentage of that is free. You're able to delete samples in the data app. So if you ever do get close to filling it, we will start giving you warnings when you get to levels that we have deemed as, worthy of warning you. So you get 3 warnings, kind of 3 strikes. Once you get the last warning, you won't be able to save more data. You'll still be able to count, you just won't be able to save the pictures. But this is a great way to see when you need to go into the data app and start clearing out, purging old files that maybe aren't useful anymore. The device has Wi-Fi. So you can see I'm on a Wi-Fi network. You're able to connect quite easily with your Wi-Fi password. There is an Ethernet port on the back. I don't have that plugged in because I'm using the Wi-Fi, but Ethernet is also a great option for really consistent connections. You can set up a network folder. So this is just a simple SMB share to a lab PC or a computer that's on the same network as the device. But it's a good option if you don't want to do email or you don't want to do USB drive. You can set up a share folder on your computer, and you can set up, the path and the password and everything here to just export straight from the CellDrop to your computer. Network printing allows you to add printers on the network, as I mentioned. And then the email setup is here. We have a technical note covering this on our website. So if you do want to configure email addresses, and I'll give the CellDrop the ability to send emails to you of data, there's a whole note about how to do that and all the settings and how they need to be adjusted. So I recommend going into that if you need help with that. Otherwise, the general system info is here. So things that might be important if you ever do ask for help at DeNovix, the most important things you can tell me are your serial number and what software you're running, so the app version. Those two are very critical to giving me a good understanding of how I can help you. But other than that, the device is pretty simple. So I've mentioned that we're happy to do these sessions at any point with any lab as they need help. So instead of making it a very, me doing all the talking and showing everything, we can do a session where that group has an iPad or a cell phone set up and they're showing me or another one of my colleagues what they're having trouble with, and we can kind of walk through and do this. It's not just because we're demoing for you. We always offer this as part of our support, being able to hop on Zoom and provide assistance quickly.